The photosynthetic pathways are differentiated into C3 pathways and C4 pathways. It was once thought that all green plants fix carbon dioxide through Calvin's cycle only. That certain plants fix carbon dioxide in a different photosynthetic mechanism called C4 pathway. Hatch and Slack observed that 4C compounds such as oxaloacetic acid, malate, and aspartate were the first formed compounds when the leaves of sugarcane were exposed to carbon dioxide 14 for 1 second. So, sugarcane is an example for C4 plant. When the leaves of rice plants are exposed to carbon dioxide 14, 3C compound called phosphoglyceric acid is formed. So, rice plant is an example for C3 plants. In C3 plants, photosynthesis occurs only in mesophyll cells. In light reactions, ATP and NADPH2 are produced and oxygen is released as a byproduct. Carbon dioxide is reduced to carbohydrates by dark reactions. In C3 plants, both light reactions and dark reactions occur in mesophyll cells, whereas in C4 plants, the mechanism of photosynthesis requires two types of photosynthetic cells. Photosynthesis takes place in two different kinds of cells, the mesophyll cells and bundle sheath cells. The C4 plants contain dimorphic chloroplasts, that is, chloroplasts in mesophyll cells are granule, that is with grana, whereas in bundle sheath, chloroplasts are agranule, that is without grana. The presence of two types of cells leads to the segregation of photosynthetic work, that is, light reactions and dark reactions, separately. Hatch-Slack pathway involves two carboxylation reactions. One takes place in chloroplast of mesophyll cells and another in chloroplast of bundle sheath cells. The first step involves two carboxylation reactions. One takes place in the chloroplast of mesophyll cells and another in the chloroplast of bundle sheath cells. Phosphoenol pyruvic acid plus carbon dioxide plus water gives oxaloacetic acid plus phosphoric acid. Oxaloacetic acid is converted into aspartic acid by the enzyme transaminase, or it may be reduced to malic acid by NADP plus specific malate dehydrogenase. Oxaloacetic acid plus ammonia in presence of transaminase gives aspartic acid. Oxaloacetic acid plus NADPH2 in the presence of NADP plus specific malate dehydrogenase forms malic acid plus NADP plus. Malic acid or aspartic acid formed in the chloroplast of mesophyll cells is transferred to the chloroplast of bundle sheath, where it is decarboxylated to form carbon dioxide and pyruvic acid in the presence of NADP plus specific malic enzyme. Malic acid plus NADP in the presence of NADP specific malic enzyme forms carbon dioxide plus pyruvic acid plus NADPH2. Now, second carboxylation occurs in chloroplast of bundle sheath cells. Ribulose biphosphate accepts carbon dioxide produced in third step in the presence of RUBP carboxylase and yields 3-phosphoglyceric acid. Some of the 3-phosphoglyceric acid molecules are utilized to produce sucrose and starch, while remaining PGA molecules are used for the regeneration of RUBP. RUBP plus carbon dioxide in the presence of RUBP carboxylase forms 2 moles 3-phosphoglyceric acid. The pyruvic acid produced in step 3 is transferred to the chloroplast of mesophyll cells where it is phosphorylated to regenerate phosphophenol pyruvic acid. This reaction is catalyzed by pyruvate kinase in the presence of magnesium 2 plus. Pyruvic acid plus ATP plus PI in the presence of magnesium 2 plus or pyruvic kinase gives phosphoenol pyruvic acid plus AMP plus PPI, that is pyrophosphate. AMP plus ATP in presence of adenylate kinase forms 2-ADP. The AMP is phosphorylated by ATP in the presence of adenylate kinase to form 2 molecules of ADP. C4 plants are photosynthetically more efficient than C3 plants because the net requirement of ATP and NADPH2 for the fixation of one molecule of carbon dioxide is considerably lower in C4 plants than in C3 plants. Let us know the differences between C3 and C4 photosynthetic pathways. In C3 pathway, photosynthesis occurs in mesophyll cells. In C4 pathway, photosynthesis occurs in mesophyll and bundle sheath cells. In C3 pathway, the carbon dioxide molecule acceptor is RUBP. In C4 pathway, the carbon dioxide acceptor molecule is phosphoenol pyruvate. In C3 pathway, the first stable product is a 3C compound called 3PGA. In C4 pathway, the first stable product is a 4C compound called OAA. In C3 pathway, photorespiration rate is high and it leads to the loss of fixed carbon dioxide. It decreases carbon dioxide fixation rate. In C4 pathway, photorespiration is negligible and it is almost absent. Hence, it increases carbon dioxide fixation rate. In C3 pathway, the optimum temperature is 20 to 25 degrees Celsius. In C4 pathway, the optimum temperature is 30 to 45 degrees Celsius. Examples of C3 plants are rice, wheat, and potato. Examples of C4 plants are maize, sugarcane, tribulus, and amaranthus.